sir has joined, sir. So
Yeah.
we'll go live. Just I will.
Yeah. Now I request our HOD to Kiran sir. You can talk to Dr. Pooja. She's my 1 year senior, and you could talk to her. And she wants to know whether is there any opportunity after while during the this lockdown
Sir,
period to give any online
sir,
classes
sorry to interrupt, sir. We
through
are on live now.
class.
Sir, sorry to interrupt. We
Yeah,
are
yeah,
going
no,
live now.
yeah, no problem, no problem.
Sorry, we can discuss. Yeah. Yes, sir. We shall start, sir. Now it's 7.
Yeah.
Divaga sir.
Hey, sir. Hey, sir.
Sir, participants are yet to join. Shall we wait or we can start? I think only 3 of us are there.
No, in
No, sir,
YouTube.
they're joining in
It's
YouTube,
streamed
sir.
through YouTube.
Yeah. Sorry, sir.
Only-
Sorry. I think participants will join through YouTube.
Yeah, they joined.
Yes,
okay,
sir.
thank you,
Already
thank you.
50 plus they joined, sir. Yes, sir.
Okay. Thank you, sir. Thank you.
So you can check YouTube also.
Yes, sir. They have joined already.
Divagar sir, can you tell me YouTube link? I have to tell me www.
I have. No, sir. It will be big links. So just I have sent you a mail. Just check it, sir.
Yeah.
Or else I have sent it through WhatsApp also.
Yeah, YouTube. Okay.
Yeah, we'll start.
Yeah, thank you. And you can talk to her, and she wants.
Sir, we shall start now.
Yeah. You
Yes,
can.
sir.
Yeah, you can start.
HOD sir. Thank you. Very good evening, one and all. Joined us for this event. I am Divakar, one of the coordinator of this program. On behalf of Department of Biotechnology, Sri Venkateswara College of Engineering, I welcome all the participants for this international webinar event. One important lesson that COVID-19 has taught us is that we cannot rely only on the healthcare systems. It is a responsibility of our immune system to tackle this pandemic. Yes, it's our responsibility to know about and improve the immune system. Today we have Dr. Pooja Shivashankar with us to deliver a talk on complement immunity and vasculature. Before this, I request convener of this webinar, Professor E. Nakiran sir, Head Department of Biotechnology, SVCE, to deliver the welcome address for this program. Sir, over to you, sir.
Thank you, sir. Good evening to one and all. First of all, I would like to thank the resource person, Dr. Pooja Shivashankar, Department of Immunology and Autoimmune Diseases, University of Molecular Medicine, USA, to accept our invitation and deliver the international webinar on complement immunity and vasculature.
One second, sir.
Welcome.
Voice, they are telling, the participants are telling voice is not audible.
Oh,
in YouTube.
sir, I think it will have some lag time, sir.
Yeah. Now it's okay. Yeah.
Yeah.
Now it's okay.
So can I start, sir?
Yeah. Now
Yes, sir,
it
you
is
can
okay.
proceed, sir. It's fine, sir.
Yeah, now it is okay.
Okay, sir. Thank you. Good evening. Good evening to one and all. First of all, I would like to thank Dr. Pooja Shivashankar, PhD instructor, Department of Immunology and Autoimmune Diseases, Institute of Molecular Medicine, UT Health. Institute of Molecular Medicine, UT Health, USA, for accepting our invitation to deliver the international webinar talk on complement immunity and vasculature on today's session. And also, I welcome all the participants of this webinar to get benefit out of this guest lecture. I feel this is the need of an hour because to overcome with the pandemic, as Divagar said, it is very important to boost our immunity, especially the complement immunity and vasculature. So I request all the participants to interact with the speaker and get benefit out of the immunology as well as the vasculature. And I welcome all the participants, and I thank the organizing secretary Dr. Prabhu and the coordinator Dr. Divakar as well as Ratna, and also I express my gratitude to the management secretary sir and the principal for accepting our invitation and giving permission to conduct this webinar. With this, I warmly welcome all the participants, and also I welcome the resource person to deliver the guest lecture. Once again, I welcome you all, and be interactive and be interactive with the resource person and get benefit out of the webinar. Thank you.
Thank you so much, sir, for the nice welcome address. Now I request Professor Prabhu, sir, to introduce today's guest speaker.
Yeah, thank you, Dr. Divakar. Before introducing today's speaker, I would like to share my personal friendship with Dr. Pooja Shivashankar because she is my 1 year senior when we were doing our PG program and MPhil program in University of Madras, AC Tech, Department of Biochemistry, University, India, Tamil Nadu, Chennai. And she always, whenever we go to canteen for taking tea and all, she would be sitting in the library, and always she would be taking notes, keep on taking. That time e-textbooks are not available as now.
That's right.
Yeah, so she always she would be taking notes from the textbook, and that also nowadays many Indian books are available, whereas during that time and all only we have foreign textbooks, so it is available only in the library. So she would go always there, and she would take all the notes. So that too not brief notes, she would be taking all detailed notes, and she would be transferring that notes also to other students and their juniors. That was very useful for everyone, even including me. So all type of subjects she was very excellent, and she was that time itself I could see her academic power and academic cleverness on the different type of subjects, and she likes taking classes very well. That time itself, when we were doing our MSc program, she would come and take classes for PG students when she was doing PhD. So in that way, she is very close to me, and she's also good in her research. So I want to discuss, explain about her education, and she completed her BSc Bachelor Science in Chemistry, University of Madras in 1991, and 93 she did her MSc Biochemistry, University of Madras, and in 1995 she did her MPhil Master in Philosophy Biochemistry in AC Tech, University of Madras, and 2003 she completed her PhD Biochemistry and Molecular Biology in 2003, and she was working as visiting scientist to complete PhD graduation, Department of NMR, All India Institute of Medical Science, New Delhi, India. And afterwards, she after completing her PhD degree, she worked as assistant professor in the Department of Biochemistry, Professor Dhanabal, and she taught biochemistry, bioorganic chemistry, and microbiology, signal transduction education to undergraduate and postgraduate students. And 2019 to 2010 to 11, she was working as a teaching assistant for MBBS MD students, first year students of University of Texas, because she moved to US that time, and she settled in Texas, and she took classes for MD students, Health Science Center at San Antonio, Texas, taught medical microbiology, and she had her postdoctoral training experience from 2004 to 2006, and she worked in the Department of Geriatrics, University of Texas Health Science Center at San Antonio, Texas, and she from 2013 onwards, she had a more responsibility of online instruction through Canvas, and she also got some proposal grant for different type of project proposals and protocols, and she also wrote she has written many writing responsibility grant proposal for many type of project work, and also she got honored and awards. She was selected as top 10 posters for cutting edge research in podium speech for highlights Gulf Coast Vascular Research Consortium, Houston methodologist and faculty research seminar in internal medicine divisional seminar series at UTMD Anderson Cancer Center, Houston, Texas, and she also invited speaker on role of dietary risk factors in intestinal cell proliferation and metabolomics approach experimental mice, and American Thoracic Society. She is selected as a best paper in the category of in vivo pulmonary fibrosis model study, and she also invited in 2012 for giving talk on molecular aspects of cell senescence and age-related diseases, and she is also editorial position for different type of journal, World Journal of Hepatology and World Journal of Gastroenterology, and elsewhere journal publication group, Biointerfaces, Colloids and Surfaces, and Journal of Pharmacy and Pharmacology, and Frontiers of Cellular and Infection Microbiology, and she is also serving to the community as a judging committee member for Junior Senior Academy of Science, as well as she is a certified health coach for Houston community people, and besides research with her experience, she is also teaching instructor and certified health coach for Wellness Institute of Texas Center, and she has more publications, more than 1, 2, 3. I'll wait 1 minute because I can't read everything, so otherwise we'll be missing her talk. 25, more than 25 international publications she has, and she has published proceedings in conferences, more than 8, and she has written invited articles in 2 different journals, and she published 3 book chapters in international books, Wiley Publication, Blackwell publisher, as well as Kim Stoll publisher. So with this brief introduction, I am very much happy to invite our today's speaker, Dr. Pooja Shivasankar, for giving the talk related to complement immunity and vasculature, and I hand over the mic to today coordinator, Dr. Divakar, for next process.
Thank you, sir, for the introduction. Now I request today's guest speaker, Pooja Shivasankar ma'am, to share your screen so that I am hitting my screen so that you can able to present.
Okay.
Your screen. Yes.
Can you see?
Not
No.
No?
yes, ma'am.
Okay, I think it is on now. It's live.
Yeah, correct. It's shown.
See that?
Yeah,
Yes,
correct.
In
So yeah, that's correct. Yeah. No, thank you so much. Thank you so much. This is such an incredible introduction. An honor for me to present my science to you guys, to the audience of SVC and global audience. We have our colleagues who have joined us from Norway, and we have our colleagues joined from New Delhi, and I also have my colleagues from United States who have joined me right now and watching me right now presenting this seminar. And I give a declaration along with my title, which is Complement Immune System and Vasculature, as Dr. Nakiran introduced. This is the need of the hour. Thank you, Dr. Nakiran, for inviting me for this presentation. I feel so home, return, and I feel so much virtually present in Chennai. And thank you so much to our coordinator, Dr. Divakar, who was here right from 8:10 and connecting to all these, virtual devices. And more essentially, I would like to thank Sukumaran Prabhu, who is my friend. I still remember going, out as a batch of 21 students to watch Titanic movie, Titanic movie, then Jurassic Park. So what a wonderful time we had in that, year or in those study time periods. Wonderful, wonderful experiences we had. And I still miss those days. But here I am today with you guys virtually and to present my field of study. So another declaration that I would like to give is it's an educational presentation, and all the authors who have contributed to this research have no conflict of interest whatsoever. And the research was carried out in Brown Foundation Indian Institute of Molecular Medicine Research Center for Immunology and Autoimmune Diseases, and it is affiliated to McGovern Medical School, University of Texas Health Science Center at Houston, Texas. And to begin, yes, we have always overlooked complement immunity, thinking that it is just there to complement the innate and adaptive immunity. But whatsoever, there is a very pivotal, integral aspect of complement immunity to the innate and adaptive immune system. And the complement immunity, as you all can see, is active is active with the three different cascade pathways, and the name tells that this is the most classical form of complement cascade, which is the classical pathway, which gets activated by any sort of antigen antibody interaction, and which recognizes the complement 1 factor. So if I have to put here as complement 1 factor, then it's going to take a lot of space. So I'm shortening it to C1, C2, C3, and C9, which is the actual cascade of a series of these activation steps involving 9 of those complement proteins, whereas we have almost nearly 30, more than 30 actually complement proteins, which take part in both innate immune system and adaptive immune system. And the fact that antigen-antibody interaction is such an important non-specific host response to any kind of stimulus that we get either from the self, that is our own cells, which are toxic or which are damaged, or the pathogens that enter through the environmental assaults, can activate the complement classical pathway. So the C1q is the one which goes and recognizes antigen antibody, and it clubs with the other two subunits, which is the R and S, so to activate further the downstream cascade proteins, including, of course, the C1, C2, and C4. All these activations lead to the C3 convertase enzyme trigger, and that C3 convertase is now going to take the lead of breaking down C3 to C3b and C3a. At this point, there is another pathway that converges with the classical pathway upon the opportunistic infections, is the lectin pathway. And the lectin pathway is recognized by mannose binding lectin proteins of the host to the manosugar on the pathogenic cell membrane carbohydrate moieties, or even the self antigenic stimuli with the mannose binding ability can activate further down the serine proteases, which can trigger the lysis of C3 by the C3 convertase. All the three pathways, if I have to mention the last one, which is the alternative complement pathway, is the spontaneous pathway for C3 hydrolysis. They all converge at the level of C3 convertase. This is a sort of very basal threshold trigger that every system, physiological system, for us to say metazoans or animalia, have this in place, so that no host cells can trigger or exacerbate inflammation in any way whatsoever. So although it is involved in pathogen recognition too, we have the self aspect of the spontaneous hydrolysis of C3 involved in most of the host inflammatory responses, which is the dangerous one. So, and this is the culprit where we see all the autoimmune conditions and physiological distress that can lead to chronic inflammatory degenerative diseases. So all these converge at the level of C3 convertase, thereby activating the cleavage of C3b and C3a. And now C3b, along with its other cofactors, is going to activate C5 convertase, so that you can cleave the C5 factor, which is the complement 5 factor, to break down it into C5b and C5a. I'm purposefully waiting on to the C3a and C5a to go on a very roller coaster ride with you all for the next 45 minutes. So let me complete the pathway of the complement cascade, making the membrane attack complex, which involves polymeric organization of C5b, C6, C7, C8, and many multimers of C9 together to give you this barrel-shaped membrane attack complex, which can lyse eventually the pathogen or the damaged cells that are causing the inflammation in the host system. So in chronic inflammation, if I have to point out on this, is because there is exacerbation and a dysregulated activation of alternative complement pathway, so that you get to release lots and lots of these anaphylatoxins C3a and C5a. Just like any other cytokine, they have their own receptors on all the myeloid and non-myeloid cells, and they start signaling and triggering the chronic inflammatory response in the host. And as you see over here, these are 7 transmembrane spanning domain receptors, and they have for C5a, you have C5AR1. I'm designating as 1 as a is a very recent nomenclature, because we have
Pooja,
found...
1 second. Divagar, it is not only the complement in the first screen is visible in YouTube. Shall we give?
No, sir, now it is fine, sir. No, sir. Now it is fine, sir. Now it is fine, sir. We resolved it, sir.
Okay. Okay. Thank you.
Yes, sir. Yes, sir.
Okay.
You can proceed.
Okay.
Hello.
So can I continue? Vascular injury, and you have pancreatitis injury. This is the most studied one, extensively studied one in terms of complement immune system impacting the vasculature. Okay, so these are all the result of unrestricted activation of alternative complement pathway. And all we know so far is how leukocytes interact with endothelial cells. Ideally, they respond to the immune cell chemotaxis by cytokine release and chemokine release, and macrophages in the inactive form or the very naive form as monocytes are going to get activated at this level and get infiltrated through ICAM-VCAM interaction. Although recently we have more factors like P-selectin from the endothelial cells, which can attach and interact with the monocytes through P-selectin binding ligand and activate monocytes, and then macrophages can cross the vascular barrier. And in the way it also stimulates angiogenesis and smooth muscle activation, and eventually leading to tissue injury. In terms of adaptive immune response, there is not much that is known for complement and aflatoxins interplay with the vasculature and adaptive immune cells, which are nothing but the B and T cells. And although debatably there are reports that lymphoblasts like B lymphoblasts do express some C3AR, and they can bind to C3A, and C3AR axis can release antibodies through plasma cells, because B cells get activated and differentiated. Although this is a debatable phenomenon, what is well known is the activation of the adaptive immune response by the antigen presenting cells, which are the dendritic cells or the macrophages. The dendritic cells, and as we know that we have C3A, which is a very highly expressed or highly cleaved and aflatoxin, but its response is very much at threshold, and the culprit is mostly the C5AR. And what it does is, in the dendritic cells, C3A, C3AR signaling expands the Th1 cell proliferation and subtype is skewing, whereas there is also a parallel Th2 cell proliferation that releases IL-10, and IL-10 goes on to compact and contract the face of Th1 by inhibiting proliferation of Th1, whereas C5AR does not allow that to happen. All it does is it polarizes the cells to Th1 population, and the expansion of this Th1 population is what is the foundation of all the chronic inflammatory diseases, and it is the most studied and most advanced emerging field in inflammatory bowel disease, which is now emerging as a serious condition even in youngsters ranging in the age of young adults ranging in the age of 20s or mid-30s. And the other detrimental effect of C5A through C5AR is on the macrophages, which induces the ERK signaling, so that there is chronic inflammatory mediators such as IL-6, IL-8, and interferon beta response, and even you have IL-1 release, TNF release, and which exacerbates inflammation. But nature has a check on this macrophages by expressing another receptor for C5A, which is the C5L2. Now it is known as C5AR1 and C5AR2. So the C5AR2 is kind of a sequestering receptor, so that it does not allow C5AR to bind to C5AR1 to bind to C5A. But as we say that always the bad, the evil empowers the good one. So the C5AR1 has a greater affinity for C5A. So no matter what kind of check you put on, C5AR1 does the job it wants to do. So the knowledge that we have is now the free-floating aflatoxins are going to interact with the antigen-presenting cells, which kind of educate your B and T naive cells, and therefore you have an adaptive immune response that follows the innate immune response. But our question was, how about endothelial cells? We all know that endothelial cells do interact with macrophages and dendritic cells and eosinophils and neutrophils, but how about the naive lymphoblastic phenotypes of lymphocytes? And so to check on this, what we said was, okay, let us now stimulate endothelial cells and see if they have the ability to interact with the T cells and B cells and help them activate and differentiate and polarize in the system. So we started with the in vitro studies, and we used two cell lines, human vascular endothelial cells, primary human vascular endothelial cells, and we also used human lymphatic, dermal lymphatic endothelial cells. But the data that I'm presenting today is the vascular endothelial cells, because we're mostly talking about the vasculature. Although vasculature means you have the lymphatic vasculature as well as the blood blood vasculature going hand in hand in all the exchanges, and keeping the immune cell repertoire in check and surveillance. We also used human primary bone marrow mononuclear cells as a pool of both T and B lymphoblasts before the T lymphoblast get dispatched to the thymus for differentiation and activation, whereas our B lymphoblasts get matured and they get differentiated and activated in the spleen and other secondary lymphoid tissues such as lymph nodes, peripheral lymph nodes. We also used two lymphoblast cells by nature, and those were C1RB7 and MOL4 to see if endothelial cells which are stimulated with anaphylotoxins can augment activation and polarization of these cells. And all the experiments we used purified anaphylotoxins, not the recombinant ones, to simulate human situation. If we are in this place of the study, what will happen to us when our endothelial cells are stimulated with anaphylotoxins. And the speciality of the study also goes with high serum media, because most of the time when an antigenic stimuli is given, stimulus is given in vitro studies, scientists tend to use serum-free media so that you can trigger and sensitize cells. But that is not what we have in our system. Our system has all the serum component and everything inside, although we heat inactivated the serum so that we don't have the complement factors that come from the bovine blood. So we had the high serum media with heat inactivation, and we show here that HUVEC cells do express both C3AR and C5AR, and they recruit our stimulus or the antigens that we add to this media, C3A and C5A, as little as 50 nanomolar, and you know how little that range is. And sometimes in my studies further down the slides, I show C3A being used as 500 nanomolar also, and you would see no change because of the saturation of those receptors that work at the level of threshold. And we do have a very robust response of C5A, as we know that C5A is more potent than C3A, and it does that for both C3AR and C5AR1. And also you have to appreciate that C3A preferentially upregulates C5AR1 expression, and C5A preferentially upregulates C3AR. And this is one kind of inquisitive we have now to see. And this has been proven by European group also, who came to present, and they said, oh, we see that all the time. Do you see any reason for that? And we say, yeah, we are working on it. So let's work on it later. But today I'm going to present to you the promotive activity of the C3AR mRNA gene, as well as the promotive activity of C5AR gene. Promoter activity of C3AR is controlled by two transcription factors, H1 and CJUN, whereas nuclear factor YA1 is the one that transcriptionally activates the expression of C5AR1. Both are increased in C3A and C5A stimulation. There's also another transcription factors which was delineated last year, 2019, which tells us that the STAT3 pathway also can take up the promotive activity for expression of C5AR, and that is the reason why we have chronic inflammation mediated by C5AR1 signaling axis. Okay. And also we show here that C5A stimulates very high amount as well as copious amounts of recruitment on the cell surface for C3AR and C5AR1 receptors, and live cells also showed very high expression of expression as well as recruitment on the cell membrane in the flow cytometric analysis. Next, we question if there is any change in the endothelial characteristics, because we are seeing a responsiveness of endothelial cells to these C3A and C5A. So we did a Boyden chamber assay of migration, wherein we have the endothelial cells seeded onto the top layer of the trans wells, and we put the stimulus, which is the C3A and C5A, and we incubate them for 24 24 hours, and we stain the cells which have transmigrated to the bottom surface of the trans well by swabbing out all those which are remaining on the top surface of the trans well. So what you see over here is those cells which have actually transmigrated to the bottom surface. And as you see over here, C3a, you can compare that C3a and C5a. There's more transmigration of endothelial cells. And also what you can appreciate is the cell surface area that has increased in the C3a and C5a, which is very much important for angiogenesis and neointima formation when there is vascular integrity being challenged. And we see that both C3 and C5a could augment that transmigration as well as the cell area, cell surface area enlargement. And this was very important to know that with the cell surface area expansion as well as the transmigration, what we lose is that the barrier integrity. Now that endothelial cells have started moving, you must be hearing about this a lot because these cells are now moving and they're inducing all the interstitium to go and pass the clotting mechanism augmented in COVID-19 patients. This is how it happens when you have the vasculature opening up. Platelet factors, platelet activating factors do get expressed, and they start coagulation reaction or coagulation cascade as your preventive and protective mechanism. And most of these COVID-19 patients can be seen having like coffee beans like clots in their body. And if not, they have micro clots in their venous circulation, risking them with deep vein thrombosis and pulmonary embolism, sometimes even stroke and cardiac arrest. So barrier integrity, we see there is a decrease in both C3a and C5a. As early as by 4 hours, you can start to see the decrease. Close to significance, however, but by 24 hours, we do have very significant movement of these endothelial cells to the bottom surface, jeopardizing the barrier integrity. And the next question was, when they are moving and when they are getting activated and neointima is in the making, what happens to the B and T cells if it is considered to be present in the lumen when these changes are undergoing in the human situation? So what we did is we added B and T lymphoblasts to the endothelial cell cultures to see how they get activated, and you can see very well that the even if I have to highlight this very, very red with the red filter on the microscope, I do not see any staining of the first marker, which is the Fas ligand marker on the endothelial cells. Whereas when we have C3 stimulation, and all those cells which are B cells, the smaller nuclei are the B cells which were added to the culture of your endothelial cells, they start to express Fas-L when they come near the periphery of endothelial cells, suggesting that C3a stimulated and C5a stimulated, more so in C5a stimulated cells, you have activation of B cells when they are in the vicinity of in the endothelial cells that is stimulated by C3a and C5a. And we checked that and confirmed by means of having independent B cell cultures and co-cultures with UX cells. Also, there is a tendency of these lymphoblasts, when they are inactive and naive, they are in suspension, and when they get activated, they adhere to the bottom of the surface of your culture dish. So we started invariably looking at both the adhered population and the floating population, and what you see over here is, yes, you do have the co-cultured adhered population expressing more of the Fas-L on the B cell surface as compared to the independent B cells which were stimulated in the absence of endothelial cells. So that tells us that C3 and C5a receptor axis may be absent or inactive in the naive lymphoblasts. cells, and we checked it with the human bone marrow cells, which are a mixed milieu of all the mononuclear cells, including the monocytes, but not the polymorphonuclear cells, the neutrophils. And we see here that there is increased expression of FASL on the co-cultured cells, not the independent ones. We looked at other markers, which are interleukin receptor 1, which is a tremendous activation marker for B cells. And we also looked at the CD69, another classic B cell activation marker, being augmented by the stimulation of C3 and C5a in the presence of UVEC co-culture. And same with the human bone marrow mononuclear cells, we do see those receptors being increased in the presence of UVEC cells stimulated with C3 and C5a. So if I am saying C3 and C5 stimulated UVEC cells, that means that I have stimulated them for 2 hours prior to adding these mononuclear cells. But then the question comes, what if within the 24 hours we have the C3AR and C5AR responses on the the poorly and quiescently differentiated B cells? So we wanted to negate that aspect, and then we saw that profoundly the cytokines that are produced by the UVEC cells are the ones which lead to this activation of B cells by doing this paracrine assay. And we also used the inhibitors for the downstream signaling pathway that leads to the secretion of IL-1 alpha, IL-6 and IL-8, the MAP kinases and STAT3 inhibitors, and they significantly reduce the activation of both IL-1 receptor 1 and CD69, suggesting that it is very essential to have the UVECs activated and the inflammatory mediators required for activation of B cells in turn. But the MOLT4 have a different characteristics. MOLT4, whether in the active form or in the naive form, they're in suspension. So these suspension cultures, we do add to the UVECs after stimulating with C3A for a couple of hours, C3A and C5A, and then we leave it for 24 hours and spin down these cells into, using the spin columns and collecting those cells directly onto the glass slides. And those cells can be stained and visualized for their activation and polarization. As you see here, CD4 positive cells is the selection of T helper cells, and CCR5 is the marker for Th1 subtype, and this is also a receptor for RANTES, which is another inflammatory cytokine which augments and polarizes T helper cells into the pro-inflammatory Th1 subtype. And whereas the Th2 marker we used is the interferon gamma receptor 1. You can see interferon gamma receptor 1 is very essential to control and induce the CD8 toxicity pathway, so that you have killing of those pathogenic insulted or pathogenic cells or infected host cells. But with C3A and C5A, we do not see much of the change in the Th2, but there is fireballs like appearance of the Th1 polarized cells. And we did see a very profound distribution of mononuclear cells of human bone marrow, and we see that both C3 and C5A augments CCR5 positive cells, whereas there is non-significant change in the Th2 population. We also confirmed that with the polarization, there is activation of the IL-2 receptor in the presence of C3 and C5A stimulated endothelial cells, not only in cell numbers, but also in the intensity of the expression levels of on each cell membrane was very, very significantly high. And it has been shown to, we proved that it is being attributed to the endothelial cytokines IL-12, P70 and IL-27, which leads to the T helper 1 polarization. So the cytokines are essential for the activation as well as the polarization of T helper response. So with this summary, with this portion of my study, I would like to summarize with this little cartoon showing that not only that vasculature is responsive to C3A and C5AR, but they are also capable of activating and differentiating B cells and activating and polarizing T cells before they are transmigrated into interstitial tissue space. And the second question was what happens in vivo, and if this response is through the C5A-R1 axis or C3A-R1 axis. But for this presentation, I'm just giving you the data of the C5A-R1 axis, where we have used the knockout mice to causatively relate the inflammatory sequence in the lymphoid tissues. And this paper is already in the submission process, and but the partial 2 abstracts dividing these complete data has been published as abstracts in Molecular Immunology with my presentation in 2016 and 2018. So let's give a little description about secondary lymphoid organs. As we say over here that we do have many lymphoid tissues, secondary lymphoid tissues. Besides the spleen, we have almost 450 lymph nodes. In a mouse, if you see, you have only 22 lymph nodes, but those are enough considering the size of the animal. And it also includes the mesenteric lymph nodes and Peyer's patches. And this is the structure of your lymph node that you can see having a bulb-like structure, like kidney bean-like structure with a concave surface and a convex surface. And the convex surface is the one that receives the lymphatic fluid, which is quite similar to our blood, except that it has very less plasma proteins, and it also has less of it does not have any RBCs. So anytime we draw the lymphatic vessels, we draw green in color, and we draw the arteries in red color, and we draw the venules in blue color. Okay, so you do have the afferent lymphatic vessels that bring in the immune cells, antigen presenting cells, and the waste and the draining lymph into the lymph nodes. And you have the plumpy endothelial sinuses which bring in naive B and T cells from spleen and thymus so that they can harbor into their own follicles. And there is a paracorticular space and a parafollicular space for T cell colonization, and we have very cell follicles within the cortex area. The middle region mostly has the veins and arteries for distribution of nutrients and exchange of carbon dioxide and waste from the entire tissue parenchyma, and that is taken by the big axis or the hilum, which is connected to the major draining blood vessels. And you have the after the exchange of the lymph draining, and even the educated B and T cells, even the activated B and T cells leave the lymph nodes through the efferent lymphatics so that they all can go get assimilated at the level of the lungs, pulmonary duct, where the new blood cells or the purified blood cells come up and get oxygenated, and then there is also an exchange of all the immune cells which have been educated, activated, so that you have a robust immune response in your systemic circulation when the blood leaves the cardiac system. So apart from B and T cell follicles, you do have a few of the memory B and T cells, so that you have, if you are subjected to an environmental assault for the second time, the memory B and T cells can respond robustly and immediately, rather than going into the bone marrow and bringing the memory B and T cells. Most of the T cells are debatably, in the memory form of the T cells are debatably present in the bone marrow, flow. So because B cells are the ones which mostly occupy the spleen and the lymphoid tissues. And although there is more accumulation of T cells, naive T cells in the lymphoid follicles. Apart from this, yes, the afferent lymphatic fluid brings in the dendritic cells, neutrophils, macrophages, and eosinophils. And based on what antigen presentation cells are presenting and activating these B and T lymphocytes are given as eosinophilic response or neutrophilic response. And you do have these vasculature, lymphatic endothelial cells and vascular endothelial cells. And then there is also reticular fibroblast that hold the tissue parenchyma. So what we did is, let's see what happens in these lymphoid tissues when there is chronic inflammation. What happens is the Th1, as we have shown, and the Th17 in the case of allergic and hypersensitive inflammatory diseases, there is an imbalance with the greater polarization of Th1 and Th17 versus Th2 and Tregs, which control these two guys. And we asked if C5a can lead to this Th1 polarization in vivo in the via the lymphoid endothelial cells. So this part of the study, we are going to more focus on the lymphoid endothelial cells. And the first thing we did was the lymphatic flow. This is a very, very important aspect of any research who deal with obesity, lymphedema, and lymphedema-associated cardiovascular complications, lymphedema-associated B lymphoma and cancers, because there is always obstruction of lymph fluid in these, very, I should say, sluggish human lifestyle. When there is retention of this edema in or the lymph fluid in their lower extremities, and that obstruction leads to activation of autoimmune conditions, because your body because the cells have to keep moving in the lymphatic flow. When there is obstruction because of your sedative lifestyle, then what happens is you are going to the sedentary style of your body is going to feel as if these cells are kept for no reason, and they start to recognize them as self-destructing cells, and they start to attack your joints and the lymphatic sinuses, including the your sinus joint sinuses. Okay. So that's why many people who are edematous tend to develop rheumatoid arthritis and other lymphatic associated diseases. And what you see over here is an acute response of C5a stimulation by 4 hours, which obstructs the lymphatic flow in the inguinal lymph node area. So the injection of the dye, which is ICG, is given at the base of the tail, and once it crosses the lymphatic plexuses, those you can see as a network or a meshwork of vessels, they pass through the lymph node, which is the inguinal lymph node. And once through this inguinal lymph node, there is only one pathway, and there's all unidirectional, as we all know, blood as well as the lymph flow in unidirectional pathway, and it goes to the coaxial lymph node, from where it joins into the pulmonary duct for exchange with the purified blood. And we could take out the frames to track one of those propulsion impulse of the lymphatic flow, and we saw that how it travels very nicely, neatly. You can see there is another propulsion or the impulse or the lymphatic vascular contraction that has started to begin, and you get another one when you pass the period of 4.16 seconds. So we are measuring this in terms of seconds actually, and as we see over here, wild type animals, when they are stimulated with C5a, so it is an intradermal challenge, so that we are inducing the dermal lymphatic endothelial cells, and which is absent in the case of C5aR knockout, suggesting that C5a or C5a axis is involved in endothelial activation and dilation of the dilation of the lymphatic vessels. Here I want to show you guys a video, if I have How do I show this video? I think, yeah, it started. You can see the baseline pulsing of the lymphatic vessels. Once through the lymphatic, the meshwork is very clean. The lymph node is very much accepting the unidirectional flow, and it is also sending upward. So the mouse is inverted actually. So, and here it is the C5A stimulated mouse. See how the dye is stuck in a very reverberating fashion with the backflow inside the mesh. And also you can notice that the lymph node is enlarging because there is obstruction of the flow. I will be playing this video again one more time so that we all get an idea. And but C5AR1 knockout mice, they do show some reverberation as well as backflow. You can see this flow from coming from here, but the lymph nodes were having very easy flow through of the lymph. And the C5AR knockout mice, again, receiving the C5A, have a very robust flow of the lymph through this inguinal node. Area. So I would like to go, and without talking, you can observe the changes. So let me go. Yeah. Just appreciate the magnitude of the flow. So not only the flow was obstructed, but we also saw some remodeling of the lymphoid tissues. And when we injected C5A at every 24 hours interval for 7 days, we saw that by day 3 there is more lymphoid cells or BNT cells recruitment, and there is also hypertrophic changes. There's budding of the lymph nodes with more enlarged BNT cells for cell follicles, and as maximum as we could go to day 7. And we have other studies where we did the lymphedema model, and we went for 14 days of time period, but I'm not discussing that data today here. And but it is very much appreciative that the lymph node enlargement is one of the very important aspects of C5A detrimental effects. And not only that, we do have inflammatory mediators such as C5AR1 INOS, which is an inducible nitric oxide synthase, which goes high in comparison to the endothelial physiological NOS, ENOS, and is high, and and the IL-6 and RANTES are high at the level of day 3 when we start to see the changes in the lymphatic follicular recruitments. And but we couldn't see the obvious changes in the lymphatic structure or the cellular population in the knockout mice while given 7 days of C5A intradermally. Although we see that also we see that there is very poor distribution of the follicles in the C5AR knockout mice. So generally reasoning that there is something wrong in the absence of C5AR1, you are not able to mediate this inflammatory response, and that tells us that how potent and how inflammatory, how detrimental C5A, C5AR1 signaling could be B. And we do see a very significant decrease in the expression of the all inflammatory markers. And we do also notice that the INAS is present on the subcapsular lymphatic vessels, the afferent lymphatic vessels being activated with inflammation, and therefore there is INAS expression. And also there are lymphatic sinuses which are very high in expression of INAS, whereas you can see here this is the high endothelial venule, which is the plumpy venules bringing in all the knife cells, and that is not actively expressing INAS. So only the lymphatic endothelial cells are inflamed and activated due to the C5a intradermal challenge, suggesting that yes, endothelial cells do show responsiveness to C5a inflammatory trigger. And we also see that CCR5 positive cells occupy the paracortex area as well as the afferent lymphatics. That means there is draining and mixing of the active T cells in the afferent lymphatic itself, and to give you a sense of Th1 polarization before they are egressed from the lymphatic tissues. So the process of homing and aggression is very, very important and attributed to the robust response of your adaptive immunity. And as we see over here, including the chronic inflammatory responses, where we see that active T cells occupy the paracortical area, and they are activated before even they leave the efferent lymphatic flow. To confirm that it is because of the mouse endothelial cells, we did a harvest of the endothelial cells by magnetic column selection with CD31 antibody-coated beads. And we used C5a, and we also use C5a inhibitor and L-nil inhibitor, which is a specific inhibitor to INAS. And we added mouse endothelial cells after 1 hour and left for co-culturing and stimulation for another 24 hours. The next morning, we took out the activated, the so-called differentiated immune cells, and we took out the cells for RNA expressions, and we took out the supernatants to see the INAS activity in terms of nitrite levels. And we see here that a very neat reduction in the Th1 polarization could be seen by concentration-dependent exposure of or countering of C5a effect with the C5a inhibitor. And we also saw with the classic INAS inhibitor very much almost, I should say 50% from the C5a, you have a reduction in the expression of CCR5. Therefore decreased polarization, and that was attributed to the that was attributed to the STAT3 signaling, because C5AR levels did not decrease with MAP kinase inhibitor, but C5AR was blatantly low at the in the STAT3 inhibitor exposed endothelial cells. This tells us that yes, STAT3, we actually prove here the recent publication that a STAT3 promoter activation is required for C5AR1 expression. Also, we do have a very sound decrease in the INAS level, and so therefore C5A activated or C5AR axis adapts the STAT3 as the downstream signaling pathway preferentially, although you do see some reduction because of the cytokines that are produced and that also activate through autocrine fashion all the MAP kinase inducible cytokines, which are IL-8 or TNF or IL-1. And then we did all these cytokines, but for time consideration, I did not add all those data over here. But I want to show you an essential part of this investigation, which is the p-STAT3 nuclear localization. And as we see over here, there is very beautiful localization of STAT3 in the nucleus. and whereas the translocation is inhibited by the STAT3 inhibitor, and we also use a positive control, IL-6, which is augmented by the STAT3 downstream signaling pathway, and you can see 100% actually being translocated into the nuclear periphery, nuclear area, whereas inhibiting could bring down all the STAT3 activation. This summarizes that yes, whatever we saw in vitro studies, we did see and confirm through the lymphatic endothelial cells in vivo, and it does it through the INAS dependent Th1 polarization, and that INAS is not from any reticular cells or any other area, but very much confined to the lymphatic endothelial vessels. The third and the final aspect of complement immune system, very important with respect to COVID-19. Why we do have this, very diverse response in different population and different individuals. Although there are many news you can hear, oh, this guy is so healthy, he is the fitness freak, but still he got pneumonia, his lungs are filled and he can't walk. And at the same time, there are people who are walking with lungs, half-filled lungs, with the fluid, and still they do not show any symptom of distress, and that is because of balance in their homeostasis, and that balance occurs through circadian rhythmicity. And here again, I would like to declare that authors have no conflict of interest, and this whole data is submitted to a grant application this June, and manuscript I am just now preparing. Okay, so normally a human has a health triad, and that health triad is 3 partners: light and dark cycle, good nutrition, and very good physical activity status of your body. In the light and dark, you have 2 other aspects underlying. The light brings in the sunlight, which is very important. So sunlight exposure for your body's homeostasis is very, very important. And the dark brings you sleep, and which is very, very important for your hormonal homeostasis. So this tells us that human being, if I have to put the time period of light cycle or activity cycle, it is from 6 a.m. to 6 p.m. So the Zeitgeber time, that's what we call those in the circadian nomenclature, how your body changes to the circadian rhythm with the dark and light cycle. So we start with ZT0, which is 12 a.m., which is your midnight, and we go on to go to 6 a.m. And from 6 a.m., we come to, which is the ZT6 essentially, and then we come down to ZT12, and from ZT12 to midnight, we go into the ZT12 phase. So this phase is called ZT0 phase, and this phase is called ZT12 phase, when we are actually very, very active. And the 6 a.m. to 6 p.m. is the light cycle, and 6 p.m. to 6 a.m. is considered as the dark cycle. Mind you all that mice have they're nocturnal. So whatever we have at ZT0 is to be considered as ZT12. Not considered, it is the fact, because they're nocturnal, they're awake in the night and they're sleeping in the daytime. So all our injections that we gave in the previous in vivo study that I shared with you were at around 6 p.m. So I would go to the animal house, inject the animals, and I come and then I see a huge, lymphoid tissue. If you go ahead and open an animal and see and try to locate inguinal lymph node, you won't be able to see that also. It will be so translucent and very small. But when I injected C5A for 7 days, and when I cut open the animal, you can see that huge new, lymph node. That tells us how much inflammation and how much of lymphoid follicular recruitment it has taken into with the C5A administration. And so I was like, okay, what will happen if I inject the animals at 6 a.m.? So I did this study with the concept of circadian rhythmicity, and that is how I got this review article also published last month. So you do have you're very high, as I already shared with you, the ZT12. What I would like you guys to pay attention to is the ZT0. You do see some follicular recruitment, but the size of the tissue, the remodeling has not occurred. More so in the C5AR1 knockout, there's no change, and very meager population of B and T cell follicles, and it tries to grow in the ZT12, like how I showed you previously also, but there is very ill-defined follicular arrangements as we noticed previously. And what is very important is, this is the second specific aim of our grant proposal, where we show how intestinal inflammation can also be impacted by C5A. And ZT0, sometimes, your C5AR1 knockout mice, you don't even see the lymph nodes. So I wanted to include this to let the reviewers know that how appreciative we should be of the phenotype of C5AR1, that you don't have any kind of protection. Also, that tells us that you have a dual-edged sword in the C5A, C5AR signaling axis. And by ZT12, do you see this? One node can become like this hyperproliferative B-cell lymphoid follicles. And wonderful analysis we had in this. And we took those lymph nodes. And for this study, I'm sharing only the inguinal lymph node profiling. We did the transcriptome array profiling, where we saw 9 upregulated genes and 11 downregulated genes among 66, which remain without any variation. And we do see some very important, I point out to integrin alpha 5 is because the third specific aim involves cardiac toxicity, and where integrin alpha is also reproducibly downregulated significantly in cardiac tissues. And very important, the cadherin 5, another adherence, because if you guys remember, vascular endothelial cells showed the barrier integrity being challenged and jeopardized. We see cadherin 5, all those which are involved in the adherence of and the adherence as well as the intact luminal intima of endothelial cells in the vasculature, both in the because now we are isolating endothelial cells from the entire tissue, so we cannot negate the blood vessels as well as the lymphatic vessels. So you have cells from both blood vessel and endothelial blood vessels as well as the lymphatic vessels. And what you have to notice is the IL-3, which is the marker of neointima or angiogenesis. It's like a growth factor, and it increases the growth, as well as there is also studies showing that blood vessels, endothelial cells, can be converted functionally to become lymphatic endothelial cells because of the growth stimulatory effect of IL-3. And also what we show over here is the NAS2 and NAS3, which were not in the plate. We added, after confirming all these through individual RT-PCRs, we added the RT-PCR profiles of NAS2 and NAS3 to appreciate how there is a twofold increase in the NAS2, which is the inflammatory NAS, compared to the endothelial NAS, showing that endothelial cell integrity as well as health is jeopardized with the exposure of C5A through intradermal challenge. And we presented the gene data into integrative genome analysis software to know what are the putative biomarkers of vascular disease. And these are the ones that we can think of even in the COVID-19 patients or any kind of viral or chronic pathogenic disease. And those genes that have an interplay in vascular diseases are regulation at the step of regulation of angiogenesis, at the step of regulation of vascular development, the neointima, and the mononuclear cell migration, which is very important with respect to our lymphocytes, because if we put the cell migration and infiltration, we put the mnemonic in America. It is called "Never Let Monkeys Eat eat banana. So we want to show how essential this path of cellular recruitment, immune cell recruitment at the site of inflammation or infection. Never, N stands for neutrophil. Never let second cell that comes to the site of inflammation or infection is lymphocyte. Never let monkeys. The third one is the monocyte, and the monkeys eat. The fourth one is the eosinophils. Banana, the B is the last one, basophils. So you can just appreciate here that basophils, among all the five essential mononuclear cell migration, basophils are the ones which have to come in the last. But here in chronic inflammation, there is so much of basophilic cells present that can uncontrollably activate alternative complement pathway, releasing this potent destructor in our system, leading to chronic inflammatory diseases such as all the vascular diseases, whether it is a degenerative disease, neurodegenerative disease, stroke, or pulmonary embolism, or any kind. Once you have the angiogenesis and vascular development pathways in jeopardy, all you are going to start is with the protective mechanism of blood coagulation. And now you can see people are draining from the autopsies of COVID-19 patients, dead bodies, like coffee beans, the blood is seen in even kidneys. Here in UT Health, all the anatomy department.
Pooja, sorry to interfere. Now the Indian time is 8:30. Participants are asking feedback link. So how many slides are there?
It is the end. I am showing the control here.
Okay. Thank you.
Mechanism. No problem. The control mechanisms that run through the homeostasis of circadian system have three loops: loop 1, loop 2, and loop 3. And we are looking and studying all the three loops pathways, and the pathways that contain and control the complement regulations involve most of the complement regulatory proteins and circadian regulatory proteins. Whereas in the case of oxidative stress, there is a toppling of these control mechanisms, leading to extended period of activation of the complement pathway as well as chronic inflammation. All that you see is not so bad, because there are many endogenous factors which can control, but in chronic inflammation we do need to take help of the pharmaceutics that are available either in practice or in trials, which are shown in the red blocking arrows, can be controlled and try to reverse, but it is not possible, as we all know. It goes and ends into multiple organ failure. Yes, to summarize here, it is the intradermal challenge at ZT0 that contributes to diurnal changes and toxicity, and there is also endothelial activation and inflammation in the peripheral nodes, and putative biomarkers are hinting towards neointima development, blood clotting as precursors of tissue injury. And these markers are now in our laboratory envisaged for potential therapeutic interventions in arthritis-induced cardiac inflammation. I just informed you that. That is the summary, and I would like to thank my chairman, Dr. Rick Wetzel, and my colleague, Dr. Stacy Mueller-Ortiz, and my technician, who's Idang Lee, and our collaborators with the Metabolic and Degenerative Diseases Department, who are dealing with the Circadian Rhythm Project with us, and we are all as multi-PI grant, we have submitted this, and Christine is the lab leader, and Bahran is her postdoc. Thank you guys, and I'll be happy to take any questions that you may have.
Yeah. Thank you, Dr. Pooja. You have given detailed information about the complement.
Thank you so much, ma'am.
Okay, so now I call upon Dr. Divakar, coordinator, and Dr. Ratna, Miss Ratna. You can ask the questions from both from YouTube as well as from this Gmeet.
Yeah, now questions are presented, sir. So I request Ms. Ratna ma'am to moderate the questions.
Thank you so much. Thank you so much. Thank you so much, Ratna. Yeah, yes. Even to immunology field, maybe you would have heard like how people tend to, use different drastic conditions to just get your immune response in cells. So, yeah, but we were lucky to have complement immune as
And
our.
because our previous head of the department now is secretary of the college, Dr.
I see.
Shivanandam sir, he is a specialist in immunology, and
Oh,
now he has established a immunology lab in our college department.
I see. I see.
So many people are doing immunology project under him also. So we are very much interested in immunology, and he was in US for 15 years, and he was doing immunology related research work, and he developed one vaccine also. So okay, so without having detailed time to expand all this, I hand over the mic to the Ratna coordinators, Ratna as well as Dr. Divakar.
Yes. Yeah. Yeah. Oh yeah, there will be many antigens which come from the antigenic determinants of host cells or as well as any pathogen. So pathogenic determinants can also augment this adaptive immunity. So that's the whole point, right? When we put never let monkeys eat banana, all those lymphocytes are very naive actually. Okay, so every trigger, every pathogenic assault is going to induce a set of B and T cells, and now they're going to create that memory repertoire and store it for the second assault, right? So you do have many, but C5a and C3a are going to take care of your surveillance. So whenever you have the life of your neutrophils, for example, I got infected with corona. My lungs are filled with pneumonia because I have a lot of neutrophils storm, and those neutrophil storm is going to bring in my T cells, and they're going to stay. The neutrophils have to stay. Ideally, the period for neutrophils to stay in the site of inflammation or infection is only 18 hours. And after 18 hours, they have to vacate that place because they're not going to be there forever. And now the T cells come over, and because T cells have antigenic response now, you have a cytotoxic lineage proliferation, which is CD8. And also you have, along with CD8, you have natural killer T cells. So you come with those as non-specific targets. At the same time, you have the B cells bringing in the plasma cells, so that you have antigenic antibody neutralization process in the place. So if you put those, I think we have many other host factors that can involve, not just the C3a and C5a, because complement starts with that first antigen-antibody interaction, which occurs at the non-specific stimulus level. We have C3 and C5a, and to tell you that, hey, come, exacerbate this inflammation. Hey, come in, basophils, you also come in, and eosinophils, you also come in, so that you are actually, you want to create that media kind of a response. Hey, see, I am standing here. I have this red flag here. So C3 and C5 will do this job, and your blood cells are like, your endothelial cells are like, oh yeah, I have to process this, expedite this response, and send all these inflammatory cells to the inflamed area or the infected area. That's how C3 and C5 have become important to bring in the adaptive immune system. No problem. I hope so. Oh, that's a very good question. There is an electrode set that we use based on the cell membrane area of endothelial cells. Normally, if you see, when we see the what is it called? the epithelial cells and the endothelial cells, they're the same, right? Character-wise, they are squamous cell epithelial cells, squamous cell endothelial cells. The only difference we call those as endo and epi is one is on the surfaces, one is on the internal luminal surfaces. Okay, they are also facing the surface only, but it is the luminal surface. So when you have these cells, they have electrode potential to added to it. So there is an instrument that is called ohm meter, OHM, ohm, which is the unit, right? So ohm meter, you can use those, and it has two tongues attached to these electrodes. Okay, what you do is you put one tongue into the transwell, and the other tongue you put it into the plain media in the bottom. Okay, so now that plain media is going to measure all the electrical current that is carried by the membrane potential of those transmigrated cells, and the top tongue is going to measure those cells which are remaining in the upper surface of the transwell. So now you can measure the current, and the current will vary as you see the movement of the these cells to the bottom surface. You start to see the reduction in the electrical resistance because there is gaps between these cells. And also what if I didn't have time to very nicely explain you guys the transcriptome profiling, where we see these the tight junction proteins are also disturbed, and we can relate that to our endothelial transmigration assay results. Oh, wow, very good. Anaphylatoxins, I just informed in the beginning slides, maybe you missed and you joined late. Anaphylatoxins, the name itself tells you that they give you an anaphylaxis-like response, right? In anaphylaxis, what happens? You have hypersensitivity, you have histamine release, you have IgE-mediated degranulation, right? That is anaphylaxis response that happens through any food poison or any maybe even snake bite. But what happens is what happens with the anaphylatoxins is they are not majoritively produced by eosinophils, because anaphylaxis response is ideally considered to be eosinophil mediated, and it has IgE mediated degranulation. Whereas in the case of anaphylatoxins anaphylaxis like response, you have what is that called, non-IgE mediated degranulation or cell lysis, and there is histamine release and anaphylaxis that can give you inflammation. That's why the name anaphylatoxin. I think I